Okay, so welcome to this next video in which we are discussing type 2 activation of endothelial cells. Okay, so we're in the process of looking at the different types of blood vessel that you're going to find within your tissue which has been infected with this Staphylococcus aureus. So you'll find arterioles, which are these small blood vessels which have a smooth muscle layer. That's the important thing. Of all of this structure, these other layers that we've drawn, they're kind of trivial and they'll be very, very thin if they're present at all in arterioles. They're far more important in arteries, but you will have them to some extent, okay? And the important concept to take from this is that you've got an endothelial cell layer sitting on the basement membrane with smooth muscle cells surrounding it. That's what's important about arterioles. They have smooth muscle cells still. Smooth muscle cell layer still. Okay, so now let's move on to capillaries, and I'll just label this up as an arteriole. Okay, so this is an arteriole. Right, okay, so let's now look at the structure of a capillary. So capillaries come next. Now capillaries are tiny, simple little blood vessels compared to the arterioles. Okay, so capillaries are a tiny, tiny little tube. They are literally big enough for a single red blood cell to squeeze through them, so they are very, very small. Okay, so the structure of a capillary therefore looks like this. It only takes a single endothelial cell usually to make the entire circumference of the capillary. So here is this single endothelial cell making up the entire circumference of the capillary, and this is the lumen down the middle here. Okay, so again this endothelial cell is sitting on a basement membrane which mainly consists of collagen. So I'll draw this in turquoise here. Whoops. Okay, so that is all the capillary consists of. It's got this basement membrane which is holding the capillary in shape. Basically it defines the shape of the capillary, and then sitting on the basement membrane you have these endothelial cells, and then you have a tiny little tube down the middle of this structure. And of course you have to remember this is a cross section. The real thing will be a three-dimensional pipe, okay, going off like that. So then you'll have a tiny little tube going down the middle, and that will be barely thick enough for a single red blood cell to move through. So it's a very small little structure, and you don't have any more layers, so it's just an endothelium with a basement membrane. Now venules are actually very similar. They're just effectively bigger capillaries. Okay, so we'll now draw a venule. Okay, so venules, again, what you'll have is just an endothelium and a basement membrane. The difference is that they are bigger blood vessels, so they take more endothelial cells to line them than simply one endothelial cell. Okay, so here come the endothelial cells. Okay, and even though I haven't actually drawn the whole structure that much bigger, I hope that by the way that I'm drawing these endothelial cells and the fact that it's taking so many of them to surround the circumference of the venule, it will be obvious that this is a much bigger blood vessel than the capillary was. Okay, so here are our endothelial cells. Let me give them nuclei. So here's a nucleus, here's a nucleus. Continuation. Okay, so there's our endothelial cell there, and again, all it's sitting on is a basement membrane of mainly collagen, which gives the whole thing structure, because remember, the endothelial cells are not particularly rigid, they're a gooey mush, okay? So they need something that's actually keeping this capillary in shape, and this is this hard basement membrane. This almost is like the foundations for the structure. Okay, so a venule, I would encourage you to think of it just as a bigger capillary. It's got more endothelial cells surrounding it than a capillary, so it's got a much bigger lumen. Many red blood cells can fit through this. But it is just an endothelium with a basement membrane. So basically, many capillaries reconverge to make this bigger venule. And one of the primary targets of the inflammatory response isn't actually the capillaries, instead it's these venules. So capillaries will be affected by the inflammatory response, but venules are a big player in the inflammatory response. And in fact, all three of these types of blood vessel are going to be really important in their own way in the inflammatory response, which is why we've gone through their structure. Right, okay, so we've now got the tools we need. We've got the arterioles, the capillary bed, and the venules. What I now want to do is talk you through the basics of type 1 activation. I'm going to summarize what happens in type 1 activation, and then we're going to see how type 1 activation is a very short-term response. It lasts for about 10 to 20 minutes, okay? However, the decent thing about it is that it occurs very quickly. So the Staphylococcus aureus has arrived in our skin, let's say, and basically you will be able to induce type 1 activation of endothelial cells and the portion of the inflammatory response which it causes in minutes. Basically, it's very, very quick. Right, so what happens is certain molecules, certain pro-inflammatory molecules arrive at the endothelial cells. Now, the archetypal pro-inflammatory molecule that causes type 2 activation is a molecule known as histamine, and this is released by mast cells. And I realised that that cell just looks like a normal Staphylococcus aureus cell. And I slightly regret I've drawn this now, because this cell should be a lot bigger than the Staphylococcus aureus cells, because these are little prokaryotic cells, whereas this is a massive, great eukaryotic cell. So imagine that it's bigger, okay? And this is what's known as a mast cell. So all over your body, you have this cell type known as a mast cell. Okay, now when the mast cells get danger signals, so when the Staphylococcus aureus is present, there will be all sorts of danger signals coming to this mast cell. Okay, and the mast cell is going to release histamine in response to these danger signals. Okay, so when the infectious agent is present, mast cells are going to release histamine. Now they have histamine stored within them in vesicles, so they're absolutely stuffed full of vesicles. So let me make one of these the nucleus, just to make it look more like a cell. But the other ones now, the other two here, they are vesicles, and these vesicles are full with histamine, which I'll colour in green. Okay, so when the mast cell senses danger, what it's going to do is it's going to start releasing histamine. Okay, and this histamine is an utterly prototype of a type 1 activating ligand, basically. So what it's going to do is it's going to go to the three types of blood vessel that we have within our tissue. It's going to go to the arterioles, the capillaries, and the venules, and it's going to act on the endothelial cells of these blood vessels. Now it's going to act on receptors that are on the basolateral membrane of these endothelial cells. So let's have our endothelial cell drawn here. Okay, so on the basolateral membrane you have receptors for histamine, which are G protein coupled receptors. So here we have our 7 transmembrane G protein coupled receptor, and for histamine, it's the H1 receptor. However, there are other molecules, other pro-inflammatory molecules, which are capable of inducing type 1 activation. But histamine's kind of like the prototype. Okay, so histamine will bind to the H1 receptor, and all of these G protein coupled receptors that respond to pro-inflammatory type 1 activating ligands are GQ coupled. This is going to activate the GQ pathway. Okay, now I'm not going to go into the full details, but basically what's going to happen is the GQ pathway is going to cause calcium to rise within the cytoplasm of this cell. And this causes many things. So in all of these endothelial cells, this is happening. So you're getting this type 1 activation in all of these endothelial cells. So in the arterioles, the capillaries, and the venules. Now, one of the things it leads to, and I'm just going to put that this is going to cause type 1 activation. Okay. Now, one of the things this leads to is it leads to the endothelial cells producing pro-inflammatory molecules. So it's going to produce prostacyclin, or prostaglandin I2, and it's also going to lead to the production of nitric oxide. Okay, now these two molecules are going to particularly be important in the arterioles, because in the venules and the capillaries we know that we don't have any smooth muscle cells surrounding the endothelium, so the endothelium producing vasorelaxant molecules isn't that important. Okay, now you can imagine that if the endothelial cells here and here are producing vasorelaxant molecules, then the tissue will be soon filling up with these vasorelaxant molecules, and they may well diffuse back to the arterioles. You can also imagine that there may well be other arterioles that are leading to other capillary beds that may well be very near to the to the capillaries and venules of this arteriole here. Okay, so the venules and capillary beds producing these vasorelaxant molecules may well be important in causing relaxation of other arterioles that are near them, but it's specifically important in the arterioles, okay? So when the endothelium starts producing this prostacyclin and this nitric oxide, it's going to diffuse back to the smooth muscle cells that surround the arterioles, and it's going to cause them to relax. And when they relax, that means that they're all going to increase in length, and when they all increase in length, then the circumference of this ring of smooth muscle cells increases, and therefore you get dilatation basically, and that leads to the lumen also dilating, and that means that more blood can flow through the lumen. So all of the arterioles are going to allow more blood to flow through them, so you're going to get a greater amount of blood arriving at the affected area. Okay, now if we just go back to what I should have told you right at the beginning now, there are 5 pillars of inflammation, okay? So let me go on to the other page to talk you through these. Okay, so there are often said to be 5 pillars of inflammation, okay? And these were decided upon by years ago, basically. So a guy known as Celsus, which you shouldn't confuse with Celsius, came up with 4 of the 5 pillars of inflammation. So Celsus came up with, and they're all in some old language which I suspect is Latin, rubor, which means redness, okay, then calor, which means that the affected area becomes hot, okay, so calor, which means heat, okay, tumor, which means swelling, dolor, which means pain, and functio-lysae, which means loss of function. Oh, sorry, no. Celsus came up with these original 4, and then later another one was added, so this one is much later on. Later, functio-lysae was added, which means a loss of function. Okay, so when you get inflammation of a certain area, basically it leads to these 5 pillars of inflammation. It leads to redness of that affected area, or rubor. It leads to the affected area becoming hot to the touch, calor. It leads to the affected area swelling, tumor. And it leads to the affected area becoming painful, dolor. Okay, and finally it also leads to a loss of function of that area, which is functio-lysae. Okay, right, so in the next video what we'll do is we'll continue this discussion.